Okay, welcome. In this video, we're going to do a walkthrough of how to do BLAST on NCBI, and it will essentially be a walkthrough of how to start homework problem number 1 for Module 5. This is an assignment that's really related to a lot of the work that my lab does. What we're going to do is try to infer whether there's conserved functions by blasting Drosophila genes against Tribolium, which is the model system in my lab. And we're going to be asking this question about whether the 4 proteins that we know comprise the dosage compensation complex in Drosophila melanogaster are actually conserved in Tribolium castaneum. So just a little bit of background about why this is an interesting question. Fundamentally, the question is, how do males get away with only having 1 copy of the X chromosome? Well, it turns out in Drosophila, they get away with it by hyper-expressing the X chromosome. So if you look at the average expression of a gene on the single X in a male, it's roughly equivalent to the same gene in females where there are 2 copies of that gene, and also on average the same as genes that are expressed from autosomes. So they're hyper-expressing. In Drosophila, the protein components of the dosage compensation complex are pretty well known. And these are the 4 main ones. There are 2 RNA genes that we're not going to look at, but male-less, male-specific lethal 2, male-specific lethal 3, and males absent on the first. And so we're going to take these genes and blast them against Tribolium to find out if we have potentially conserved function, to see if Tribolium is doing dosage compensation the same way. We do know that Tribolium upregulates the X chromosome in males, but we don't know how. So for starters, we're going to go over to NCBI. So I've given the link here, and that should take you to a page like this. Blow this up a little. And what we're going to do is the first thing here where it says, well, first thing to notice is that I'm logged in as me. Yours probably says login if you've never gone to this site before, you don't have an NCBI account. You don't have to set up an account to complete the homework. It just allows you some nice features of it will save your search history, so you can go back and look at the results of searches that you've done recently, but it's not necessary. Second thing is to notice we're going to change from all databases to gene, and then we're going to search for the first gene in our list called male-less. And then you can just hit the search, and it may spin for a minute, but then you'll come to a results page. Now, why search only the gene database? The NCBI database is enormous, and if you search all databases for male-less, you are going to be overwhelmed with the number of hits. You can see already searching for male-less in just the gene database gave us 7. Okay. But if I had searched all databases, we could potentially get thousands of hits. Okay. And so then making sense of those is a challenge. So what we're trying to do is let's refine our search to exactly the database we want. We know we want to search for the male-less gene from Drosophila, and so we can start there by searching for male-less in the gene database. So we're going to sip from the fire hydrant rather than get overwhelmed by it. So if you look at your result page, you should notice that the first one here is actually in Papilio. The second one is Malus from Drosophila melanogaster. And I've actually given you the gene IDs. If you go back to the PowerPoint, these are you should use these when you're doing your searches for the homework. So, for example, I've told you that I want gene ID 35523. If you go here, you can see in the gene database that number matches. Now, another way you could do that is take the gene ID directly, and rather than search on Malus, you could search on the gene ID. If I do that, it will jump straight to the gene page in the gene database for Malus. Now, why did I not start that way? Oftentimes, if you're doing this on your own for a real project, you won't know the gene ID up front unless you've gotten that from the literature somehow. Usually you'll only know the gene name, and so usually you'll be searching with something other than the gene ID. In this case, I've given you the gene IDs. You can start there. And so that takes you directly to the gene page, and it gives you some background information about the gene. There's a ton of information here that we just don't have the time to dig into all that. I'll end this presentation just showing you some of the help specifically for BLAST. But I encourage you to explore here. There's just too much going on for what we need to do for learning BLAST. So if you scroll down to the graphical representation of the Malus gene, you'll see two lines here. The first one is refseq. And let me jump over to the slide. So I've got this highlighted in the slides. So these are really showing you the same part of the fly genome, where this is the same gene. It's just that refseq has it as one entry, and then it's also in the NCBI database under its fly-based gene number, and that's lifted over from Ensemble. Okay, I talked about this a little bit in the first video when I was introducing BLAST, how these databases mirror each other. And in principle, these will match exactly. That's not always the case, but we're going to use the refseq for everything that we do for this exercise. Okay, so now I'm back on the website. I can click on this refseq representation, and you'll see it drops down to show me the RNA and the protein that correspond to this Malus gene. So the purple are the RNAs, and the red are the proteins. And so I'm going to hover my mouse over that bottom one because I want the longest version of this gene to blast against tribolium. So you can see when I mouse over, this box pops up that has a bunch of information about this specific protein. So it's got its length here, okay? And then farther down, it's got some options. If I wanted to download the FASTA sequence for this, I could click on that link. But super conveniently, NCBI has built in the ability to take this sequence and directly send it over to the BLAST tool. So I'm just going to click that link, and you can see it's taken me over to the BLAST-P, that is the Protein BLAST website, and it's automatically filled in the accession number for that longest Malus protein isoform. Okay, and you can even see it's filled in an autofilled job title telling me that this is Malus isoform A. Isoform A happens to be the longest. Let me just catch up for a second, going back to the slides. Right, if you're following along in the slides, so these are the things I mentioned. The accession numbers do mean something. Like NP means it's a protein, NM is a messenger RNA. Sometimes you'll see NT, which is the nucleotide sequence. And then that box that popped up, right? So isoform A, I just got done mentioning. If you mouse over all of them, you'll see that it is the longest protein for maleless. We could download the FASTA, like I said, and then what I did was follow this protein blast link. Now, just a short aside here is that if you were working on another project another time, you can get to the blast tool from that original website pretty much any. The fastest way is to just click on all resources and then go to the tools. And I think on all of these menus, blast is probably listed there. When you click on that blast tool, it's going to take you to, let's actually just go try it. Show you what I'm talking about here rather than just talk about the slides. Actually, let's do it this way. Go back to where we started, when the sort of landing page for NCBI. If you go to all resources, under the tools menu, you'll see the blast website there. And you can get there under tools on almost all of these menus. It has a smart prioritization. Like if you're working on DNA and RNA tools, the most common reason or common tool you would use is blast. So anyway, that's a quick way. If you click on that, it takes you to the main blast or web page, where you can choose which kind of blast you want to do. Do I want to search nucleotide versus nucleotide? We're going to do protein. And the reason I did this as an aside now is by using the pop-up box to automatically fill in what we want to blast, we've skipped this page, right? Because we were on a protein, when we sent it to blast, it automatically filled the accession number for the Drosophila protein. Okay. So going back to the slides, right? If you click there, you'll get to here. And then what we are doing is as if we had clicked the protein blast button on that main web page. And then in our case, we could have copy and pasted that accession number in, but it automatically filled it because of the nice little pop-up link that we got when we moused over. And also notice that the database to search has been auto-filled for us. This happens to be the default when you do BLAST-P. It's the non-redundant protein sequence database, so NR. If you scroll down a little bit on the web page, you'll see the program selection. Again, just time constraints. We're not going to talk about the other alternatives. These are primarily ways to run BLAST that are better at finding sequences that are more evolutionarily distant or more diverged sequences. They'll do forms of iterative BLAST, and where it will find initial matches and then build what they call a position-specific scoring matrix. So however well it aligns, it will weight those more highly and then redo the search, so that variable regions don't really matter, but conserved regions matter a lot. and then it will search the database farther and or it will get hits farther and farther away because it knows these are the only important parts of my sequence. So that's SciBlast and PhiBlast are different algorithms for things like that. But for now, Tribolium and Drosophila are not so far apart, at least in these genes, that we have to worry about that. We will get some hits. And the question will be, how good are they? Okay, and so once you've got that chosen, you can hit run. So here I'm back over on the website. You can see that's filled in. Whoops. We're going to leave that blank for the moment. The first time you run through this, that should be blank. It just had Tribolium in there because I've done this earlier in the day. This is selected as Blast P, and then we're going to hit the blast button to run it. Note that all the parameters that I talked about and went over in the first part of the blast, the intro to sort of how blast works, you can modify those down here. Okay, so especially if you're doing a project and you're not sort of getting the results that you expect, tweaking these parameters based on all those things I talked about in the first lecture, this is where you would do that. For now, and usually on the first pass, you're just going to run blast with default parameters. Okay, so you'll hit that, and then it may take a while depending on how busy the blast servers are. But at some point, your results page will pop up. So that's what's shown on the next slide. I've got it pre-computed here. So this is the default run. And sort of what you would expect when you search the NCBI database with male-less isoform A, the best hit is itself, right? So all it did was return the best hit as itself. Now, there are a bunch of others, and you can see there are orthologs in other species like simulins, okay? And you can keep on scrolling. You'll see there are lots of hits, okay? But 100% match to itself. No surprise there. If you look at the table, you'll see some of the scoring things that we talked about in the first lecture. So the bit score, that is the max score. The difference between max score and total score, this is for, sometimes you'll get a match, remember, and it'll go below the threshold for part of the sequence. And so it will identify part of the subject sequence as a match to the query. And then there'll be a break, and then it'll match in another part. So this max score is for the best piece of this hit. The total score are all the matches on that whole piece of the subject genome or the subject gene. So total score is over the whole gene. Max score is just in the maximal segment pair. Okay, that's terminology from the other lecture. The query coverage, right? This is how much of the thing that I searched with, so in this case, Drosophila male-less, was covered by the subject. So 100% in this case. E value, we talked about that. What's the likelihood of seeing a match of this quality given the whole NCBI database in this case? And then the percent identity. So how many of the protein residues of the query and the subject match position by position in the alignment. Okay. So now, right, we're not really interested in the male-less matches in Drosophila melanogaster or any other Drosophila for that matter. So how do we, and you already, I gave it away a little bit, right? How do we further refine our search So we're not getting overwhelmed with data or blast hits that we aren't interested in. We're only interested in the stuff that's in tribolium. So, whoops. We can limit the search set to only look in tribolium castanium. Okay, so let's go back to blast P. Oh. Actually, look here, we can go back. This is the first thing. If we search with the gene ID, get to the mail list page, right? Go here. Whoops. And let's blast it. But now we're going to limit this to only search in tribolium. So that's our only change, is limit it to tribolium. Okay, and then we'll search again. That'll take some time to run. When it's done, you should get a table that looks something like this. Okay, so these are the results. And you can see that the top 3 hits here are predicted proteins. Okay. Now, for the homework, you're going to need the accession number, the E-value, the query coverage, and the percent identity. So one thing you can do is write those down here, just record them as you go. Another option is to download this description table as a CSV. So if you click that button, you will get a download. And so that was really fast. You just downloaded it to my website, or to my download folder on my local machine. And you can see it's the same. So now I've got my results in Excel format, which is super useful for lots of reasons, especially if you're interested in more than, say, just the top hit. Okay, so just to catch up where we are on the slides, right? We ran with tribolium. We have our data table. Here I made a note that in the homework I asked for the accession number of the protein. And just so you're not confused, because earlier in the slides I said that proteins start with NP. Well, if it's a predicted protein, it starts with an XP. So what does predicted protein mean? That just means that these are computational predictions that have not been confirmed by, say, RNA-seq. Or even if parts of the gene we know are expressed based on RNA-seq data, the exact intron-exon boundaries, starts and stops, may not be known. Okay. One of the other things you can look at from your results page are the graphical summary. So in this case, you can see this is the query. So my query is a little over 1,250 amino acids, and the best hit in tribolium is, so red means that its alignment or its bit score is really high, and it's covering almost the whole query, right? So this is just a graphical representation, a little bit different way to look at the data. And so one of the reasons you might want to do this is beyond the numbers that are in this results table, the pattern of matching might tell you something. Because genes are made up of exons, and exons contain domains or functional domains, the parts of the gene that actually sort of do the work in some sense. And you can have conserved functional domains in a protein that are actually a very small piece of the protein. So for example, a transcription factor binding a transcription factor may have a very conserved region that is the actual DNA binding region for that transcription factor. And that's highly conserved. But the rest of the protein is sort of telling that transcription factor where to go. So it's localization signal, or it has some other functional property also encoded in another part of that gene. That means that, yes, it's a transcription factor. It has some homology to whatever I was searching, but the whole gene is probably not doing the same function in both species. Okay, and that's relevant because that's where your level of interpretation is going to come in for whether the genes in Tribolium are have conserved function with the genes in Drosophila. And so how do you make those inferences? How do you make those decisions? Well, I'm giving you some reading to do. And so if you follow this link, it's a very good overview of how to interpret BLAST results in terms of whether genes are homologous, and if they are homologous, right? So inferring homology based on the similarity, but then even beyond that, inferring function from homology. So you've decided you found a homolog, then you're asking, okay, is it likely to have the same function? Okay. So you'll have to read through that in order to really answer, especially this last question. So the homework is to take each of those 4 genes. So the genes I gave you at the beginning. So these 4 genes. You're going to walk through what we just did. So we've done, I just showed you, if you're following along, you've done the one for male list. And then there's a quiz built in Canvas that is going to ask you to answer these 5 questions for each of those 4 genes, right? So it's worth 20 points, 1 point for each thing. So you'll give me the accession number for the best hit in Castanium. So what is that for male list? Let's go back here to our description, right? It would be this XP number right here. E value, right? So E value is right there. And query, percent identity, and then yes, no, maybe. Is there a gene with conserved function in Castanium? I'm not asking for, this is not purely subjective. This is based on the reading that I was just showing you. Okay, and that'll be due Tuesday the 7th. Before I sign off, just one more comment. I said earlier I'd show you, I think all of the NCBI web pages have this help button. If you're anywhere in BLAST, it will take you to this. And like I said, there's so much in BLAST that we can only just kind of scratch the surface here, because we're also going to spend some time learning to do BLAST on the supercomputers for homework number 2. But this has some great tutorials. It's got a deep dive into the algorithms. You can go as far down that path of really exploring the BLAST tools. Really interesting sort of computer science background. This really is the algorithm that made modern sequencing databases searchable, right? Because like I talked about in the first video, you can't do alignment on millions and millions of sequences. You have to take that sort of heuristic approach. Okay, so that's it for homework problem number 1 in the BLAST module.